need some DNA fragment. Okay, in here we use this bacteria suspension as our template. Okay, and the next main thing is we need primers. As I mentioned earlier, we do this experiment to identify the bacteria, so we have specific primer pairs for these bacteria. Okay, so then we use the primers, and in PCR reaction, you guys know we are constructing new DNA from a mother DNA fragment. Okay, so to make that fragments, we need DNTPs, that are the building blocks of the DNA. Okay, and then to do this reaction, we need a reaction buffer. Okay, and next one, the most important one is DNA polymerase. That is the enzyme that is stimulate the reaction. These things are we mainly use for our PCR protocol. Okay, and when we talk about the what are the this is the PCR process. That means we have single template DNA. Okay, then it will be separated by breaking the hydrogen bonds that between these two strands. Okay, then we have two strands.
Yes.
Okay, then using this DN, we have this DNTPs, that are the building blocks of the DNA, and with the help of the enzymes and the reaction buffer, it will make another strand, and there is a complete two template that is similar to this first one. Okay, likewise this process happen again and again, about 35 cycles. Ultimately we will get millions of copies. Okay, this is the polymerase chain reaction. Okay, the main stages of the polymerase chain reaction is first the denature. That means separate the two DNA strands. Okay, then primer annealing. Okay, and then extension of these DNA fragments. That is the main cycles of the PCR reaction. Okay, ultimately we get this amount of copies, and we can visualize that fragments using agarose gel. Okay, then we know the size of different bacteria that we are using in this experiment, and we can, after we check with the agarose gel, you can identify the different sizes of your relevant sample, and then you can identify what is your bacteria. Okay, this is the summary of this practical. Okay, today we are going to do only this part, the seeding. Okay, you guys, we are provided with the NB broths. You can, and also the bacteria, you can get 100 microliters from this suspension and seed into the NB media. Okay, and we can incubate in 37 like in the previous one, and tomorrow we can do the PCR. Okay, before coming to the class you can study more about the PCR. Do you guys have any questions?